The human digestive system. So the point of the digestive system is to break down your food. So the process of digestion breaks food molecules into usable forms. This is the winner of Nathan's hot dog eating contest last year, I think. Probably not digesting a whole bunch. I think he had 86 hot dogs or something crazy like that. Anyway, humans get energy and materials they need to live by eating. So that makes us different from plants. The organisms that we eat are made of all the stuff you learned in your organic chemistry unit. So carbohydrates, lipids, proteins, and nucleic acids. You'll find all of these on a food label, maybe not nucleic acids, but you'll find carbohydrates. It'll either say sugar or complex carbs, and lipids will usually say fat, and then the proteins will say protein. So the molecules have to be broken down into simpler substances so that we can use them. So here's an example of a polymer, and if you break it here, and here, and here, and here, you need enzymes to break these things. But when you do, it breaks them down into this thing, a nucleotide. And so that's the first step in the digestion of a nucleic acid. And so this is going to be the same sort of process for all of your different organic molecules. So nutrient overview, what are the monomers or building blocks of carbohydrates, lipids, proteins, and nucleic acids? And so you can do this on your own and then pause or pause now, do this on your own and then come back for the answers. So the carbohydrates start as polysaccharides, typically. You could eat a monosaccharide directly, but let's talk about eating a polysaccharide and digesting it. First, you'll break it into disaccharides, and then more enzymes will break it into monosaccharides. So again, polymer is the general term, and polysaccharide, that's the term for carbohydrates. So there's a polymer for proteins, it would be a polypeptide. But when we're talking about carbohydrates, the polymer is a polysaccharide. The dimer, that means two, is a disaccharide, and the monomer would be a monosaccharide. So those are all sugars. Lipid digestion. So this is fat, right? When you think about lipids, think about fat, at least for digesting. We're not going to talk about steroids and stuff. The most important thing that you digest in terms of lipids would be your steroids. Would be your triglycerides. So a triglyceride is a fat molecule, and I'm going to call it a big molecule instead of a polymer because it's made out of, if you remember, glycerol and 3 fatty acids hanging down. Not really a polymer because it's not like a little thing connected to another, to another, to another, to another, to another. It's just these 2 things connected together, glycerol and then the fatty acids. So there's 4 things altogether. Anyway, you break it up into glycerol and 3 fatty acids. If you look at proteins, the polymer, the general term is polymer. The specific term for proteins is a polypeptide. And you can break that down into a dipeptide and then into an amino acid. Too bad it's not a monopeptide, huh? You're going to actually come up with the names as we go. You'll have to come up with the names for all of these. So there's an enzyme, or actually several enzymes, that will get you from polypeptides to dipeptides, and another type or class of enzyme that will get you from dipeptide to amino acid. So for each of these types of macromolecules, the carbs, the lipids, the proteins, and the nucleic acids, you'll need to know this chart. So you'll need to know the polymer, the dimer, and the monomer for the most part, and you'll need to know the enzymes that break them down, and then what part of the body they work in. So is it the mouth or the stomach or the small intestine? Nucleic acid digestion is next. And so the nucleic acids are the polymers. You can break them into nucleotides, which are monomers. And the body actually has to go further, and it has to break the nucleotides down into the monomer components. So that would be a sugar, a phosphate, and a base. Remember, the sugar is ribose or deoxyribose. There's a phosphate group, and then the base is that nitrogenous base, ATCG or U. So let's look at the digestive process. Digestion involves enzyme-controlled hydrolysis reactions, which use water to break down these molecules. So remember we studied hydrolysis before, hydro meaning water and lysis meaning to break down. And so this is the opposite of dehydration synthesis, which you learned is putting stuff together. So we have to put water in to break stuff apart. And so here's the digestion of starch. Remember, starch is a polysaccharide, so it's a carbohydrate. And so you get this from things like potatoes and rice and wheat. And when you break this down, you'll get glucose. So here's an example of what it looks like. Here's a disaccharide. And if you add water, you could break this. The O could have one of the H's attached to it. And this carbon, right, all these bends are carbon, could have the OH attached to it. So it'll end up looking like this. You have an H here and an OH here, and those came from water. And so you have a disaccharide that got broken into a couple of monosaccharides, that and that. Digestion of a fat is the same idea. Here's glycerol, and these are the 3 different fatty acids. Remember the vocab here, a fat is a lipid, right? And specifically a fat is a triglyceride. So a triglyceride is the same thing as a fat or an oil. And when you break it down, you're breaking the bonds here and here and here, and you get glycerol, one of those, and you get 3 fatty acids. And so here's what it looks like. It says, here's the glycerol, and it's got carbon with a couple of O's sticking off, double bond to O and an O here, like this. Carbon is here. Then there's a carbon here with a double bond to an O, that's that one, and then there's, oops, I have it backwards, there's an O in the middle, like that. And then the carbon is attached to, it says 17 carbons, 1, 2, 3, 4, 5, 6, 7, I ran out of space, yay, and hydrogen's on either side all the way. So that's what the fatty acid looks like. So when you break those down, you're getting glycerol and 3 fatty acids. And remember, this is a chain of 17 carbons plus this original one. Digestion of protein, same thing. This is, well, each bend isn't really carbon. It's nitrogen, carbon, carbon with an O, nitrogen, carbon, carbon with an O, nitrogen, carbon, carbon with an O. Anyway, what you're going to do is break all of these down as well. Right here, right here. And so you're going to get these amino acids. So when you break down a protein all the way, you get amino acids. And notice the black part is the same in all of them. These blue things sticking out are called R groups. And that's just a review from before. And so here's a protein, nitrogen, carbon, carbon with an O. Here's one of the R groups, and this one they just actually did write R for that one. Here's the other one. So you add water in, which means you would break it here, and you put an OH here, and you put an H here, and that will give you your 2 separate amino acids once you break that bond and you add water. So it looks like that. And here's that nucleic acid digestion again. You're breaking it right here. Quiz time. Lipids are broken down into, pause this, and I will do it, into fatty acids and glycerol. Carbohydrates are broken down into what building blocks? That would be monosaccharides. Proteins are broken down into amino acids. Nucleic acids are broken down into nucleotides, which are then broken into sugar, phosphate, and base. Oh, did I say that? Nucleotides are broken down into sugar, phosphate, base. And we're going to stop right here.